I'm Kevin Eliceiri. I'm a co-chair of BINA and a professor here at UW-Madison, Wisconsin, and I really wish I could be there. Unfortunately, I had teaching commitments that I couldn't leave for, but I'm there in spirit, and I was attending a lot of the meeting. It was wonderful. This session is new for BINA. That one of the growing areas that we want to do in BINA is technology development, particularly building instruments to complement our open source software efforts. And this is a theme that we want to introduce at this meeting and keep growing with these great speakers. And it is one that we plan on continuing over the next year with various activities, including the next community congress that will be in the USA in Madison, Wisconsin. Will really stress this. We have several programs that will be about open hardware and hardware building. So we put together a fantastic group of speakers that are all here, either virtually or in person in the room there. And delighted they all could come. And we will start with our first speaker, Abhishek Kumar from Marine Biological Laboratory, and he's there in person. And Abhishek is a wonderful builder of microscopes and light sheet instruments and other instruments. And thanks, Abhishek, for talking.
Thanks, Kevin. Can you hear me well?
Yep, you sound great.
Wonderful. Okay, let's. Good morning, everyone. Buena dia. Good morning. Thank you all for being here. First of all, I would like to thank BINA and Lovy organizers for having me and giving me the opportunity to share our efforts and work. And I'm honored to join my colleagues and friends and in this majestic conference room. This is a great honor. I would also like to especially thank local organizers, Adan and Urini, and their team. They have been a wonderful host, and I'm kind of amazed by their hospitality and care and kindness, and it has been a wonderful experience. So thank you, thanks. So now let's get started. It's easy to be a last day and first speaker on the last day because everything I wanted to share, previous speakers already, they have shared with you all. So I'm going to repeat some of those things just to reiterate how important it is to share advanced technologies with community. And there are various ways to share it. There is no one way which works for everyone. So we all do our part and we all take different approaches. So please don't get me wrong. I'm not saying that this way is better than the other way. We need to take multiple approaches and see what works better for our community. So Alice and North already talked about Marine Biological Laboratory. We are a small institute south of Boston. We are known for hosting summer courses, including advanced microscopy courses, but also we have a strong history and tradition to build equipments and push the envelope of innovation at the MBL. And MBL is a heaven for me for building these tools because we attract biologists from all walks of life, from diverse scientific background, diversity of samples. So it's a very unique place to build a system where biologists, you build a system, next day biologists, they come, make it great. That gives you a challenge. Hello, okay. Sorry, we are back. So online people can hear me? Okay, great.
Yes.
So as a part of our initiative under current administration, we have two initiatives. One is, we call it Imaging Initiative, where the goal is to develop new technologies, invest in dissemination, training, education, and enable biology. So basically, we want to work closely with biologists and make sure that tools we are developing is suited for them. It's enabling them to do what they couldn't do otherwise. And that's our goal. And for developing technologies, we have three pillars. We need to work with biologists to identify what their needs are. As a physicist, I don't want to develop a tool for the sake of development. I want to work with my colleagues to identify what really they need. What are the bottlenecks? What are the obstacles? We need to work with folks from computer science and math background, because in today's world, it's hard to imagine any imaging technology without sophisticated image analysis. So it's very important to work closely with them. And so there are 3 pillars, and basically we work closely with each other to advance our scientific goals. We have a core facility led by Louis Kerr and Karsten. So we have a lot of commercial equipment. Sorry about that. Thanks. Yeah. Also, Rudolf and Michael Srivak, they develop imaging tools, but also we invite tool developers to bring their latest and greatest system, and we host them so that we all can work together. So there are multiple ways we try to incorporate and disseminate advanced technologies, which are developed in-house or developed elsewhere, somewhere else, and we invite those scientists to come, stay there, spend summer or few months and work with biologists. So that's a little bit about our initiative and MBL. If you look at it carefully, it has 3 things. In Hari's lab, we had developed this latest light sheet system. And I'll show you the system we had developed in lab, but Hari had this passion that this system is useful. We need to make it accessible. So we invested a lot of resources and time to develop a system, which is still not a turnkey system, but compact enough so that other labs can adopt it. The second part is training. Hari and I, we travel to India so that we can be there with those students and we can teach them. Third thing was those students, participants, they were not afraid. They wanted to get their hands dirty. And I can tell you, none of them had touched even a microscope before building this system. So it was very inspiring to us, seeing their motivation and dedication and passion for building this system. So it touches all 3 points that yes, we can translate these microscopes from our lab to other labs. We can train people, even though they don't have a formal training in optics, microscopy, still they can learn. And third thing is, we as a researcher need to go a step extra. Just beyond publishing a paper or doing our own research, we need to invest a little bit effort and time in making these things accessible so that other researchers can adopt without wasting their resources and their time. So this way we are maximizing everything. And this is a good example. And that's what I said. This video is still relevant and I get inspired every time I watch this video. And that's what I want to do. I will, in interest of time, I will move forward. This is one example. In this picture, you can see, can I use a laser pointer? Yeah. You can see Dr. Harish Shah. This system was the first generation dice beam developed in our lab at the NIH. And as you can see, that system, this system can't be used in other labs. It's more suited in physics labs where you are occupying entire optical table. But what we did is we invested resources. I mean, it was Hari's passion. He wanted to make sure that we need to do something to make the system accessible. We partnered with a small company called Applied Scientific Instrumentation. And the second generation system was developed at the MBL. We went there in summer months because we wanted to work closely with biologists so that we can get their active feedback while we were designing and building the system. And then I started this wiki page, dicebeam.org, where we started sharing protocols, control software, analysis software. And then John Daniel and Nico Sturman, they joined and they wrote a nice micromanager plugin so that people don't have to use MATLAB or other commercial softwares to control this microscope. And that really helped spreading this system. And I'm proud to say that now more than 100 labs, they are using this technology. And it's not a full commercial system, but people can buy parts from different vendors and assemble using all the protocols. And now it's a community-driven project. Now people contribute control softwares, analysis pipelines, sample mounting pipelines, sample holders, and all these things. So this project has really taken off. I'm going to share another microscope. When I started at the MBL, I wanted to see what sort of microscope I can build. And one thing I realized that at Marine Biological Laboratory, people are working with large samples. We all love confocal. So I wanted to build a confocal which can image faster. So my team, we developed this confocal system, which is a line scanning confocal. It has two views, one from top and one from below. So basically 2 microscopes put together around your sample so that you can image from 2 sides. And you can see we don't need it, but we developed this 3D CAD model so that we can easily share all the components, parts, and everything. And this is also, this system is very recyclable. The reason it's recyclable is tomorrow, if my colleagues or friends, they suggest some other ideas, I can easily recycle all these parts and implement some other ideas. So in a way, we are also maximizing our resources. I'll show you this example. This is in collaboration with Liz from Dr. Kevin Eliceiri's lab. And we looked at this growth of zebrafish embryo for almost 27 hours. So that tells that we can image large field of view at reasonable speed and get information we need. And in this case, this is as acquired data. There is no deconvolution processing or anything, because I wanted to show you what type of data we can acquire. And then further, you can enhance that. So what we have done with this system, in fact, we have collaborated, as you can see in this poster, we have looked at more than 20 different organisms. And now we have established in about a year and a half, 20 active collaborations. So in a way, we are operating like Lyon's Genelia Ames Center, because that's a very good example how to advance biology by sharing advanced techniques. But we are working with very limited resources. So, but it's a full-time job for one of my staff scientists to work with biologists and make sure that their biology and the system, they are good match for each other, and then help them do their biology. 2 labs, they are already building a replica of this system. So we have trained their graduate students and postdocs in my lab, and we have shared all the parts, control software, and they have taken this risk and building a replica of the system in their lab. Even though we haven't published this paper, but already we are sharing with our colleagues. I will move to third example. This is the turf system I had built for my own research. And as you can see, everything is on this optical table. But I realized that there is no commercial ring turf system. There are turf systems, and ring turf is in the back aperture. You illuminate in a circle. There used to be a system which they discontinued, but Nikon has eyelash, and there are some others. But we wanted to make this ring turf system cost effective and easily accessible. So I partnered with ASI, and using MEMS mirror, we basically develop this control system so that you can build a ring turf system easily. And I trained a postdoc from Amy Gladfelter's lab, and they bought these components and they built this system successfully in their lab. So dissemination doesn't necessarily need to be a full microscope. Even a small component could be useful. And a lot of times in my lab, we don't buy a laser box because it's very expensive for us and we don't have any control. Anything we want to change, anything we want to add, we can't do that. So what we do is we build this laser sled. We buy all these mirrors from AVR, Thor Labs, laser, whichever company you prefer, whatever wavelength you need, and easily you can put together in a cost-effective way, very useful laser sled. I also like because the students who come to my lab, they love playing with this laser sled. And it's a good way to train your undergraduates or graduate students and break this ice. And they start feeling comfortable handling lasers and microscopes and all. And there is nothing to lose in this case because the worst thing they can do is get your dichroic or mirror dirty, that's all. So this is a good way. So what we do is we share a 3D CAD drawing, and my good friend Brian Mellis has really taken this to next level, all these 3D models for teaching and dissemination. And this way, what you can do is you can just get this 3D model. It has all parts lists. If it's a fabricated parts list, you can get the drawing. If it's commercial part list, it will tell which company, which model, from where to get it. And without wasting your time, you can easily assemble this laser module to do what you need to do. Similarly, there is another example. For my own research, I had built this optical splitter for doing dual color and polarization. And two labs, they wanted this splitter because it's very flexible. You can replace components easily. You can tweak it to meet your needs. So what we did is we generated this model where we have listed every part. And then you can get, as you can see in this table, which part is from where, specification, whether it's a custom part or it's from some particular company. And this way you can get all these parts. So it requires some effort on our part, but by putting this little bit extra effort, it makes things easier for us, definitely, because now if I have to share with my colleagues or friends, I can send all these details, but it also saves time for them. So these are few examples, what we see personally, my lab, we see what dissemination means. So it's not only building advanced technology, but making this accessible in an equitable way so that people can access, evaluate, and benefit from that. And then if they want to go further, they can get resources. And if they want to build these systems, basically we share everything open source, parts list, design, software, everything easily we share with our colleagues. So now I will shift gear. And as Kevin started that there is a real need to get this community together and do start thinking about a working group in BINA for microscope builders. So initially, Alison North and Kevin Eliceiri, they contacted me, and it was easy to convince me. Certainly, I'm passionate about this, but hearing from Alison and Kevin, I said yes. And then they recruited two of my good friends, Martin Fisher and Brian. They are wonderful microscopists, and they know how to disseminate all these technologies. And now they are on the driver's seat, and we are looking to start this builders group. So last BINA Congress, we had a table, and after dinner, we wanted to see if people are interested, and so nobody came to this table. Then Brian came up with this idea, how about we go to Captain Kidd? Some of you, if you haven't been to MBL, Captain Kidd is 2 minutes away from our lab, and we do all science there, basically. Bartenders, probably they know more biology than me. So we put a small note here, please come find us in the Kidd. And you'll be surprised. We got about 10, 12 participants, and that's how this discussion is started. So what we are planning to do going forward. So as I said, Martin and Brian, they're leading this effort. So we want to organize events because we also want to listen from our colleagues what is really needed. We want to set up a website and online platform so that if you are developing a sample mount, or if you have some designs, lens folder, or anything, you can share easily. We want a forum for continued discussion. Beth presented yesterday very nicely about, and it has been very successful, Image SC. So we want to use something like that, but instead of reinventing a new forum, probably we are going to work with Jennifer Waters and her team and utilize microforum to start this discussion, continued discussion. We want to build a consensus. I want to see what is the easiest way to share this? Like Katrina and all they are doing for this data sharing. What type of metadata? Similarly for builders, we want to see what type of design, what type of information, what type of nomenclature people want so that they can read those things. If we can't speak same language, even though we have something to share, you may not benefit. So we want to come to a consensus and build this metadata for parts sharing, microscope sharing. So we are certainly looking for participants. And if you have any suggestions, feedbacks, if you want to participate, feel free to reach out any one of us or all of us. We couldn't have done this without Vanessa and Nikki. I don't know how, but once Alice and Kevin, they planted the seed, they really sailed through very smoothly. And where we stand today, we couldn't have done without their help. So thank you, Vanessa and Nikki. And of course, CGI. Vlad and Vanya, they are here. I don't think whatever little we have managed to do, we couldn't have done without a CGI support, because this is on top of 5 other jobs we have to do back home. So we are grateful for all this support from the CGI. So with that, I would like to thank my collaborators, certainly my lab members. I'm presenting, but they're the one doing all this work. A lot of funding agencies to provide us resources to keep our lights on in our lab and get all the parts to do these fun things. And I want to thank all of you for your attention, BINA and Labby. And teaching starts early. Like my daughter is teaching my son how to find a sample on one of my microscopes in my lab. So thank you all. Gracias.
That's wonderful.
Thank you.
I think we've got a few minutes for questions. Any questions for?
Yes, Katrina.
Can I ask a question?
I'll wait until the.
Yeah, I think we've got. Yeah, go ahead, Shalin. I think it's okay.
So thanks, Abhishek. I was curious how you look at maintaining this community online. So you mentioned image.sc and Microlist as 2 forums. And as you mentioned, both of these imaging instrumentation and the software for acquisition and analysis, they need to interact more closely together. Do you and others think that there should be just one place where imaging in general is discussed? Like, is image.sc the forum where we have these discussions?
Thanks, Shalin. Very good point. And good to see you, Shalin. So it's very important to share, but I don't have a clear answer to your question, whether shall we package everything at one platform. That might be distracting and overwhelming for a lot of people, but we may need to share links so that appropriate links and guides so that people can go to software page to get the software, go to hardware page to get the hardware. If they need to download parts, they can go to appropriate page. If they need the protocols, if they have all the parts, then we can link them to that page. So I think we need to think a little strategically how to share this information, because as you know, suddenly this becomes overwhelming, and somebody who's looking to start it can be a little daunting task.
I might be able, to add to that very quickly that, we when we formed MSSC, it was really to support open source software, given how broad an audience it is. And then when Jennifer Waters started the forum, the microforum, we really talked at length about whether there should be one, and we agreed it made sense to have two, but sister forums, so they cross list. And then we also hope that the microscopy DB efforts of BINA could also help with that. So I think those three things together really make up a nice community and cross reference, instead of having one forum, just because there are some different needs between the software and the hardware world. So
Right. I've been recently looking at some of the machine learning community pages, like Monet is an example which is a collaboration between NVIDIA and academic communities. And having the problem is simpler with software. Having all the information in one place does make it easy for us to find and access. So as we are thinking about, I think it will be good to even have an overlay website, which may be image.sc or something else, to make it very easy to link information and associate information about different aspects of imaging.
If that makes sense.
Okay. Hi, this is Katerina from UMass Medical School. As you know, we have developed this tool called Micrometa app, and I was wondering if you would be interested in collaborating to capture the hardware configurations of your microscope that you're building. I would love to help do that. So that, because this sounds like is the perfect example, for which we can actually be open about the hardware that constitute these microscopes. And we're also planning to build a database based in collaboration with Core Marketplace of microscopes and their configurations. So it would be great if we could collaborate on that.
Of course, and thank you so much. Already we got one collaborator, so it's wonderful. And my answer is yes to all your suggestions. Yes.
So I think in the interest of time and to give everybody the opportunity to give their full presentations, we'll hold the rest of the questions until the question and answer session at the end. So we'll hand it off to our next participant. Sorry, Kevin.
Yeah, our next speaker is Reto. Thanks so much, Reto. He's a pioneer in light sheet, and he'll be talking about oblique plane microscopy. Thanks, Reto.
All right. Good morning, everyone. Can you hear me?
Yes.
Yes.
Okay, that's good. I would like to thank BINA and all the organizers for hosting me and giving me the opportunity to present to you some of our recent work on a type of light sheet microscope that is called the oblique plane microscope, which we think is a powerful technology that can also scale. So we have developed OPMs that can image subcellular features and variants that can image whole model organisms. I have quite a few things to present, so I will just go straight in. So I will talk a little bit about the theory of OPM. I will talk about the extension of OPM with structured illumination to improve its resolution. I will talk about a method that allows an OPM to instantaneously form projective views, so you can look at 3D scenery from different angles in real time. And I will talk about the mesoscopic variant of OPM. So for fluorescence 3D microscopy, there are currently two main avenues how we acquire data. One is the confocal microscope as a laser scanning or spinning disk, and the other is the light sheet microscope. So the confocal microscope scans a focus spot through the sample, whereas the light sheet illuminates a plane. You probably have heard a lot about it. There are many advantages for doing light sheet in the sense that it's more efficient in acquiring in a parallelized fashion a lot of pixels, millions of pixels with modern cameras, and confining the excitation better than the confocal. Nevertheless, if you go to any core facility, you will much rather find a confocal than a light sheet. So they're still the workhorse in 3D imaging. And why is that? One reason is, at least in my opinion, that the light sheet system is often a very bespoke system that also needs customized sample mounting. For example, here you see a typical implementation and excitation illumination objective and a detection objective at 90 degrees. And somewhere in that space, you have to insert a sample, for example, in a FEP tube to hold an embryo. But if you want to change the objectives, or if you want to put, for example, a multi-well plate in there, that's not easily going to happen. So they're not very versatile, and as such, you know, a spinning disk, you can change objectives, you can put different samples on top of it, so they're just in many ways easier to adapt and use. So we think with the OPM there is a way out of this to make also the light sheets more user-friendly and more versatile. And to explain how an OPM works, we have to go a little bit into theory. Y'all can still hear me? Okay. So unbeknownst probably to many, a normal wide field microscope already forms a 3D image. So here, if this is a 3D cell, in the image space, there will be a 3D image instantaneously. However, that image is quite crappy in most places. It's best in the nominal focal plane. Outside, it will quickly become much more blurry and aberrated. And it's also stretched out. The axial magnification scales with the magnification squared. So if you have 100x, the axial magnification is 100 times larger than lateral. So it's grotesquely stretched out. So what would happen if we plug this 3D image back into a same microscope system, but in reverse? As one could imagine, it will undo some of these distortions, and you will end up with a replica, 3D replica of the sample over here. It will be diffraction limited, but at the very least, every point is imaged the same way, and it's not stretched out like that. That can be shown theoretically. Here you have to believe me. And interestingly, this also works if you switch objectives. If you take certain care, you can go from water to an air objective. And if you observe a proper magnification, if you choose the lateral magnification to the refractive index of the immersion, say here water, then the axial magnification, if you use that formula, also becomes n. So you, in this case, end up with a 1.33 stretched 3D replica, but it's at least stretched in all dimensions the same way. So you can create a distortion-free 3D image, and interestingly, you can now place an observation plane here wherever you want. You can move it, you can tilt it, and you can observe your cell without actually moving the cell or doing anything there. And this is relevant for the idea of OPM. So OPM, essentially here, this is the light sheet geometry and blue excitation sheet, exciting 2 points, and you collect the fluorescence from those points. In an OPM, you package that into a single objective. So it has enough numerical aperture that you can launch at an oblique angle the light sheet and collect the fluorescence. And that was first shown by Chris Dunsby in this setup here, where here schematically the sample is stretched out in this intermediate image space, squeezed back again, or in an undistorted format by a second objective, and then observed at an angle with a tertiary imaging system. So that allows essentially, at least at the front end, to package it into a single objective. That is a really cool idea, but for a long time, many in the community, including me, have not paid proper attention to it because we thought it's inevitably also associated with a light loss and a loss in resolution. So if you were here to detect at 90 degrees, your observation angle would be relatively small, lowering the efficiency and resolution. So you pay for a high NA objective, but you only get out micron level resolution that were at least in the first implementations. However, some time ago, Bin Yang, a talented postdoc at UCSF, thought up to here where you formed a remote image, there is actually no light loss involved if it's done properly. So if you collect here up to 60-degree angle, you can map that back and get a distortion-free, diffraction-limited focus of either point here and here. And there is no loss of light up to that point. Where the loss of light comes into play is when you actually want to observe this tilted plane, and you put another objective here at an angle. Say if this angle is 60 degree and that tilt angle is 30, then that tertiary objective would need a half opening angle of 90 degree. And such an objective does not exist. It would be infinitely large or would have zero working distance. However, what Bing Yang came up with is to switch immersion. So go here from air into water. And as you go in the denser medium, the rays are refracted towards the normal of this interface. So this light cone becomes much more manageable. And so as such, you can observe or collect even these high angles theoretically up to 90 degree, skimming here the surface, and create, if you want so, an almost impossible tertiary objective that can collect up to 90 degrees here. All right, so dealing with the water is a bit complicated. It can dry out. It's also hard, for example, here where it interferes with that objective to cut a corner out since it's a liquid. So here with my colleagues Andrew York and Alfred at the Calico Institute, the idea was to swap from water to oil for the tertiary, and instead of oil, using glass, because glass you can essentially cut, for example, here a wedge out that would collide with the secondary objective, and as such still get the same benefits of jumping to the higher refractive index. And this allows us then to build such an OPM with a high numerical aperture objective. This is how it works in action. In our current implementation, there are two galvo mirrors that scan the light sheet just like this. So the first mirror gives an angular change, the second one undoes that angular change, but you're still left with a shift. So that way you can scan the light sheet, and this is how it looks on the camera. Here we scan through a Vimentin labeled cell on the cover slip. It's a tilted view, so that's why it looks a bit shallow, but nevertheless you can scan through the cell without moving the objective here or the stage. Here this is some resolution measurements of the high NA OPM system, laterally about 300 nanometer. And here are the point spread functions of the OPM compared to a square lattice light sheet microscope and a spinning disk, also with the same high NA objective. And you can see they're quite similar. However, compared to the lattice, you just have one primary objective and unlimited access on the top. And compared to the spinning disk, you enjoy the advantages of light sheet. Here is an alternative resolution measure using Fourier ring correlation in a cell. And you can again see the raw values are around 300. And with deconvolution, it got about the square root of 2 better. So now, just to show how volumetric data is acquired, so the light sheet and the focal plane are tilted, and as they're being scanned, you essentially acquire a parallelogram shaped volume. So just when that comes up, that you're aware that it's not a rectilinear volume that is acquired. That's just by the nature of how the tilted imaging plane lies. And so with the OPM, we can image relatively fast, up to 10 Hertz volumetrically. Here, these are clad-encoded pits. It looks a bit like a turf image, what people like to do actually when they study clad-encoded pits. But in contrast to turf, we see here the entirety of the cells. So here, when these clad-encoded vesicles disappear, that doesn't mean they leave the evanescent field. It essentially means they're being internalized. So you capture essentially the entirety of the cell. And here we use this system to look at T cells attacking cancer cells in magenta. And here you can see in a movie how this cancer cells is getting attacked. Here just in red the T cell, and here the overlay. And we also created a rendering here of the surfaces where you can see color-coded the curvature. You see here the immunological synapse, and here how material is being pulled away from the cancer cell, which could be observed quite nicely. And the other beneficial aspect is the volume that we can image at the time is relatively large, so you see many interactions at the time between the T cells and the cancer cells. We also use this microscope for studying cell ruffling. Here, this is Gabriel and Felix in Gaudenz Stanwis's lab. You have used the system here on SU86 cells, and here you can see some analysis of both the curvature and the density of tractin on the surface of the cell, which then can be further analyzed, say what triggers these ruffles and so forth. But for me as a microscopist, it's nice to see that we have sufficient resolution to also look at that. 300 nanometer resolution is good for a light sheet, but in the realms of spinning disk or super resolution, a bit better would be nice. And so one idea was to combine the OPM with structural illumination. So here, the artistic rendering of the OPM, the light sheet comes out of the objective here, but we could also send two light sheets out. If you look from the side, they propagate in the same plane, but diverge from each other. And if they're coherent, then in the overlap area, they form an interference pattern, so they form a structured light sheet. So this was the idea, how to make structured light sheets, and as they emerge from the objective, they can also be rotated, which is a necessity to get isotropic resolution in SIM. You need at least 3 orientations. And when this structured light sheet plane rotates, in principle also the detection needs to rotate, because it has to be mapped onto the camera. So if this changes its azimuth, the detection would need to change that too, which however would be a bit too painful to implement in reality. And so what we invented is a rapid image rotator with galvo mirrors that send the image over a series of static mirrors, and depending which mirror is selected, this actually induces a rotation. So here we look straight into the device, and as the mirrors start scanning over the galvo mirrors scan over the static mirrors, you can see that this test target here at the end starts to rotate. Here, with the OP-SIM, as we call it, the OPM with structural illumination, a comparison on beats, here on the left just with OPM and on the right with structural illumination. So as before, we're in the range of 300 nanometer just with the OPM, and with the structural illumination, we can double it to about 150 nanometer. And that corresponds quite nicely to an instant SIM microscope, so it has similar imaging capabilities. Here, these are myosin and rod domains that form quite a nice structure. There are 2 motors and 1 rod in between, and the separation is about 150 nanometer, which can be resolved with the instant SIM, but also with our OPM with structural illumination. Here you see 2 pairs and a few others. So you get the resolving power of, say, an instant SIM, but with the lower bleaching and the higher speed potential of the OPM. The speed potential derives simply from the fact that nothing heavy moves. Neither the cell has to be scanned or the objective has to be scanned to acquire volumes. And that allows you to, for example, here, this is 1 Hertz volumetric SIM imaging of mitochondria dynamics. All right. Got to speed up just a little bit, just to confirm you all still with me, or you still hear me? All right, I hope so. So about the projection imaging, here we use a peculiar transform that we found. It had existed but has been forgotten, which is called the shear warp transform. So which simply says if you rotate an image and project it, you can find a sheared version of the image that its projection produces the same kind of line profile. It's a bit stretched out, but if you squeeze them together, they overlap quite nicely. So to rephrase it, for every rotation and projection, there is an equivalent amount of shearing that you can apply here to this image to obtain the same view. So if you consider that as a viewing direction, this sum. So you can easily verify that if you have a stack. So here, this is an OPM stack. And if we sum all the slices, you get 1 projective view. But if we shift the slices progressively and sum them per the shear warp transform, you get a different viewing angle of the cell. You could do that, but today graphical processing is so powerful that you don't need to use the shear warp transform. You can just rotate and interpolate the volume. However, what we found is you can also do that in an optical way, an analog fashion. So here the OPM scans through the cell, and we increase the frequency with which we scan through the cell. And once we reach the frequency of the camera, a projection along the scan direction appears right here. So this is really in an analog fashion. The camera just runs freely, and we adjusted the speed with which we scan through the volume. So now we again sweep through the cell, but in addition, we translate, or shear if you want so, the image as it comes in over the camera. And we also increase the speed. And once we reach the frequency of the camera, then voila, a projection of the cell appears. That's really in a single camera frame, a different view of the cell appears. And if we change the amplitude of the shearing, the rate or the amount we sweep it over the camera, we can change the viewing angle, also in accordance with the shear warp transform. And so the way we scan it over the camera is we have just two galvo mirrors in front of the camera, and they can essentially translate very rapidly the image. So at the same speed as we can sweep the focal plane or scan the light sheets through the sample, we can also move the resulting image over the camera. So this allows us to see live, for example, here the vasculature in a zebrafish from different angles. So every image in the movie is a single camera frame without any processing. So you could even look at it by eye and see different views of the scenery. It can be used for rapid phenomena, like here the blood flows through a zebrafish heart, viewed at two different angles. And so you can see how the blood cells enter the different chambers of the zebrafish heart. And here we used it in a hybrid mode. So these are cancer cells and again T cells. And here one attacks a cancer cell. And once that happens, we can go out of the projective view and actually go into a 3D acquisition locally where the interaction happens. So this is a large volume. This would be gigabytes of data if it were done in a movie. But here locally, the 3D acquisition was done in an event-driven manner. All right, gotta speed it up just a wee bit. So the last bit is, could we also do this with lower MAG, lower NA objective to image like millimeter to centimeter scales? In principle, yes, but the practical outcome is like this low NA objective, 0.28, the light sheet will come out at like a 10-degree angle. So it would be way too steep. So you could simply not catch it. And such a steep light sheet would also not fulfill the purpose of light sheet microscopy. It should just excite too much outside or above and below the detection PSF. So what we did, or the innovation we added, we add a glass prism here on top that by total internal reflection here reflects the light sheet so that it enters at 45 degrees. And to capture the fluorescence light, we add here a grating. That was an idea from the lab of Benjamin Yudkowitz, and it works in as such that the first diffraction order is designed to be normal to the grating surface, and so light of the first diffraction order is sent into the primary objective. This cannot be done by a mirror. If you think you could just place a mirror here and bend the light, the short answer is it will not help, but the grating does. It really bends over the light and sends it into the primary. So with these two innovations, we can have like a 3.5 millimeter wide field of view. The resolution is about 2.5 microns laterally and 9 axially. And also this is a side view of a volume, so it can actually be quite large. So again, this is the parallelogram that is acquired. The scan range was just maybe shy of a millimeter, but here in the Z direction, it encompasses an entire millimeter of height. So the microscope has quite impressive coverage. And here we used it to look at the vasculature in a zebrafish, a 3-day-old, and you can see fine detail. Each bright spot that's the endothelial cells are labeled, and each of these are nuclei. So you can essentially almost count all the cells that make up the blood vessels. And here we use the microscope to image the blood flow at 12 Hertz in a projective manner. So in gray, that's the vasculature, and in red, those are the blood cells that sip through. And you can again appreciate the finiteness in the zebrafish. So you could essentially go ahead and count all the cells. And you can also see here how the blood cells loop through some of these vessels. Here are the main artery and veins, and there's a lot of traffic, but here just a few blood cells at a time traverse the smaller vessels. And the resolution, both in time and space, is good enough that we can use optical flow to quantify at each position. So there will be tiny arrows that show the magnitude and the direction of the blood flow throughout the fish. With that, I come to the end. I would like to thank all the members in my lab, especially Bore and Binging, Andy and Alfred at the Calico for their pioneering work on the Snouty glass tip tertiary lens, James, who helps a lot on the theory, Kevin, a good colleague, Kylie at also the Calico Lachlan, who is really great in animating stuff, much greater than any of us in the lab with our PowerPoint or Illustrator skills, Etai, Philip, and everyone in the Danus lab and our funding. With that, I'm open to questions.
So in the interest of time, let's do one super quick question, and then we'll save the rest for the end of the open Q&A. So is there one quick question? All right, if not, we'll save it for the at the very end. Thank you so much, Reto. We'll have an open Q&A after all the talks. So
Thank you.
good. So our next speaker is Anita from Vanderbilt. She is a pioneer, not in her own research, but in leadership and community building and optics. And we're delighted to have her here presenting. So thank you so much, Anita.
Thank you, Kevin. I just want to make sure this slide is visible in presentation mode.
Yes. That's great.
Excellent. Thank you, Kevin, for inviting me. I wish I was in Mexico. I would have rather been there than here, where it's pouring rain at the moment. Unfortunately, as Kevin said, class and the short notice prevents me from being there. What I'm going to present is a little bit along the lines of Abhishek mentioned, but some that goes a little bit beyond. So please bear with me. I want to start, of course, by acknowledging all the people involved in this research, especially Brian Millis, who many of you know, and John Kozu, who's the behind-the-scenes optical engineer who does the hard work of building the components and putting them all together and aligning. And so on. The rest here on the left-hand side are the PhD students who've been involved for many of the things I'm going to talk about. We've been hearing about imaging across the scales, and we've mostly been focusing around light microscopy. Of course, electron microscopy comes into play, and most of turnkey traditional instruments are housed in core facilities, such as what we have here at Vanderbilt, the cell imaging shared resource, which are sort of follow the pay-for-use model, if you will. What makes this a little challenging, though, when you push the boundaries of what light and microscopy can do, is that core facilities are somewhat limited in what they can offer in terms of support, as well as working with the researchers, because these novel cutting-edge microscopes, such as what Reto just talked about, are not turnkey instruments and require a lot of hand-holding, both in the acquisition phase as well as in the analysis phase. So for that reason, we at Vanderbilt, along with myself and a couple of other faculty members here, sort of came up with the idea that we wanted to form what we call Biomid, and I'll explain the acronym in just a second, where our focus really was to push a problem-based approach to research, especially when it comes to microscopy. Biomid sits with the Vanderbilt Biophotonics Center. It's a center that I direct here at Vanderbilt, where we really are looking to push the boundaries of what light can do, not just only for fundamental discovery, but also clinical translation, both in medicine and biology, So our buzzword, like we like to push, is not only that it's trans-institutional, both within Vanderbilt and across the world. We're looking to transform or be transformative in what we discover and invent, and really push translation. And for those of you who may know, a significant portion of my career has been really focused on doing clinical translation, taking optical technologies and putting it to use for clinical care, whether it's diagnosis or surgical guidance. And a few years ago, Eric Betzik actually came to Vanderbilt to give a seminar, and my chancellor walked up to me and said, "Anita, why can't we do this here at Vanderbilt?" And I said, "Sure, we can do it. We can do a lot more if you give me some money," which he did, which resulted in us thinking about translating, not just to the clinician, but translating to the biologist, as Abhishek also mentioned. Obviously microscopy, we're all proponents of microscopy, and detectors that are more current and new have allowed us to provide new insight into what we can discover both about science and society. And of course, CISR provides Vanderbilt scientists anyway access to these microscopes of today. But we all want more. We've heard Reto mention this. We want smaller or bigger, faster. We want to go in vivo. We want to look in 3D. We want to look longer. And so we need to push, as many of us are doing, what light can do for us in this space. And so BioMed stands for biomedical microscopy, immersion, innovation, and discovery. So that our goal really, when we build anything, any novel instrument, is really to build two of them. One that we keep in the lab where we're constantly innovating, modifying, adjusting, and one that we make available to researchers around, at the moment, mostly to within Vanderbilt and Vanderbilt University Medical Center, and where we personalize the instrument for use for discovery, if you will. We also have educational programs where we try to immerse our students into this environment so they can learn from multiple perspectives, both the optics and photonics perspective, as well as the biomedical and biological perspective. So we build and develop these new researchers who are really experts in that intersection domain between the two. So I'll start with talking about the microscopes and then give you a little bit of foray into a couple of other things. The conventional ones that you're all aware of, the lattice light sheet microscope, which is what triggered, this area on Biomed here at Vanderbilt, where we really are focused on low toxicity and speed. But one of the challenges, of course, with this is that your sample holder and your sample size is very confined to that 0.5 millimeter cover slip, if you will, that holds everything in place. My apologies for that noise. But we've been able to do some fun things, such as looking at the cilia in the intestinal system and watch how they move, both in normal animal models, intestines, as well as in people with cancer. Of course, you just heard about oblique plane. I'm going to move through all the sort of the oblique plane planar illumination microscopy, or SOPI as we call it, where we built our first generation one. Again, this is probably our workhorse instrument. While yes, resolution is somewhat compromised, we find you can do a lot of things, especially with the intact brain or organoids, and really push even in vivo measurements of what we can see under these microscopes. And so here's a video, for example, where you can watch what's going on in ours in sort of in a vascular cancer cell migration, and watch what happens over time in a chick embryo. So here you have some specs of the SOPI, in this particular case, our first generation SOPI, which lets you, see what our temporal resolution is and what our imaging depth is, of course, depending on scattering. But pretty quickly, we realized we really needed a next generation of this instrument, which is modified to do both multiphoton and autofluorescence SOPI, which allowed us to double the resolution of the first generation instruments and do both visible as well as multiphoton excitation of the fluorophores in present. We also added some other features where we could perturb the sample, such as by using laser-induced breakdown and look at effective pressure waves on cells. A project where we were looking at neurotrauma and the biological first responders of mechanical as well as different forms of pressure waves to neuronal cells. And this instrument is probably by far our most used instrument, where we work with investigators to push what we can do. But one of the things that we have really focused on, because conventionally we can do a lot of labeled fluorescence to really visualize what's going on. However, when we don't know what we're looking for, we're sort of struck with not knowing what to label. And so we end up needing to look at label-free approaches. The neurotrauma work or the traumatic brain injury work is one example of one. The neuromodulation work we do is another example where we don't really know what is going on. We just know that something is, and we hypothesize, but we really need to look at what is changing in order to be able to extrapolate and figure out how we can understand the biological processes. So we built, this actually predates our lattice and our light sheet microscopy development. This instrument is called MANTIS, which stands for Multimodal Advanced Nonlinear and Thermal Imaging System, a complex instrument with lots of wires, as you can see, that we put together. My, sort of brainchild over a weekend that resulted with working with Thor Labs to make it into a reality, where this instrument combines 5 different, now 6 different techniques into one. We're focused on nonlinear optical interactions, and it can look at both coherent anti-Stokes Raman spectroscopy in a hyperspectral mode, so you can actually get a spectrum. To also simultaneously do stimulated Raman scattering, to do really fast imaging of Raman signals, biochemical signals that is based out of inelastic scattering. The system inherently can also do multiphoton autofluorescence as well as dye-based fluorescence. It can do second harmonic generation. And more recently, we've been able to add some fluorescence lifetime imaging as well as thermal micrography to this instrument. The platform is based off the Thor Labs Bergamo platform, if you will, where this sits on a turret that can be rotated through to do nonlinear imaging, thermal imaging, or wide field imaging, all with 1 microsecond speed and reproducibility, as well as resolution of 1 micron, or reproducibility of 1 micron. So we can rotate and bring it back in place within 1 micron of where it was. It has 2 laser systems, both a femtosecond laser as well as a picosecond laser, that both are used for a variety of different parts of the experiment. So it's point scan based, has 2 forward detection ports as well as 2 epi detection ports, has both PMTs and photodiodes, and really can do a lot of different things that we have used to our advantage. For looking at everything from, this is just static sample of a kidney section that you can do with a variety of different things, like the different dye stains that you're used to, but also to do specific bands in Raman scattering, such as the CH2 stretch, which is indicative of lipids, or the CH3 stretch, which is indicative of both lipids and proteins, and then overlay all of that to get a better picture of what's going on in the section. This is a picture of a rat sciatic nerve ex vivo. We've also done this in vivo. We can not only look at the CARS and SRS image, but you can add fluorophores to tag myelin. You can also do second harmonic generation, look at the collagen fibrils that are present. And the resolutions of these images are quite high. If you look at this and you really look, you can look at the melanin, you can really see the myelin sheets and the nodes around VA that are present within any one of these nerves, down to of a resolution, in this case, about 400 nanometers. We've done this with in vivo brain, so we can really look at the rat somatosensory cortex and see what's going on, look at edema, look at fluorescence, look at signal chain spontaneous activity, tag specific cells such as astrocytes and microglia and look at their activation, and then overlay the information we have from various modes. So I'm going to give you 2 examples, one very quickly. One that is focused on what we are interested in. My lab developed this idea of using pulsed infrared light to activate neurons. We can also inhibit them, but my current own personal research is focused on activating neurons, and we've been able to do this from everything from the rat, mouse, non-human primate, and human neurons. But we don't necessarily fully understand the mechanism of what's going on. And so we know it's a thermal gradient that causes the effect. We believe there is some form of a pressure, very minute pressure component. But what's happening to take this information and transform it from optical to thermal to this electrical signal is something, that we are still trying to understand. One of the things we discovered, though, when we were trying to do this experiment, is that the laser that's used to stimulate ends up causing thermal lensing. And so we need to do some focus compensation. Whenever the laser is turned on, here on figure B, you can kind of see that. We see that the signal essentially tends to drift because the focus of the nonlinear beam tends to drift. And so the two-photon effect is no longer at the focal plane. And so we have to do some kind of focus compensation to get that signal back so that we can actually see the effect. So we were able to, in fact, do that by doing some form of focus pre-compensation where we can move it just enough automatically and then bring everything back into the field of view. Sort of what I'm trying to show you here, sort of in effect, it's like adding a lens to bring this into field. We tested it using beads to see how well it would work. And indeed, we see that, can get the signal at the same level before and after. With the beads, you're not really going to see an IR effect. And then we started to look at the NG108 cells. And we very clearly see that you start to see lipid isomerization changes that happens only when the laser light is on, which allows us to now understand that what we perhaps are doing is we're causing some cell volume changes, but essentially loosening the lipid bilayer of these cells, which is what's allowing water to flow in, causing calcium to flow in, increasing the intracellular calcium, and therefore triggering IP3 receptor cells to then result in an action potential. So we've done a series of work to look at this, both subthreshold and at threshold, where we can actually see the signal. And we've, of course, published this work since. Another different example, another work that I do is looking at cervical remodeling during pregnancy, both in humans and in animal models. This is work from a mouse model of both looking at wild type as a normal pregnancy. The advantage of working with mice, of course, is that they have a 19-day pregnancy cycle as opposed to 9 months. This allows us to really study how things are changing with pregnancy, which is not necessarily fully understood. And so here is some data where we did some in vivo Raman spectra that you're seeing. We can measure. This is the clinical part of what I do in both normal wild type as well as two different models of preterm birth, RU486 as well as LPS. And then we extracted the cervix and looked at this with the mantis, and we could see a variety of different things. We can see the collagen fibers get disorganized as you go from a given day in terms of term versus what happened in these two preterm birth models. Instead of looking like the normal, which is the first row, they start to look more like what the term pregnant cervix looks like when you've triggered in these preterm mouse models. So we can look at a variety of different things, collagens, elastin, look at blood, look at nuclei. The collagen is done with SHG, and the rest are done with various stains, and really understand the biological processes. This is more conventional, what you're used to, where we can look at the cleared cervix that's been cleared with DII, and then, infused with DII, and then cleared for SOPI imaging, where you can really see the ectocervix and see how the vasculature changes from early parts of the pregnancy to later parts of the pregnancy, which is something that clinicians have noticed, that the cervix of a term person is more pink, but they didn't really had understood whether that just increase in blood flow, increase in the number of blood vessels, increase in the size of the blood vessels. And this is starting to show a bigger picture that you see just a lot of really small blood vessels that come into play at term in the cervix. I think we all know this. The biggest problem with doing all these kind of cool things is the sheer volume of data that you collect. When you do this, when you have to do data transfer, you have to certainly do data analysis and then try to do data storage, where, especially if you do anything clinical, you have HIPAA and on human, you got HIPAA and other things that require this data to be still sitting around for extensive periods of time. And because of the sheer amount of data, of course, you can pull out one bit of information and then a year later you go, oh, wait, I could pull something more and go back to that data. So it's not, it can't just sit somewhere in cloud. You've got to be able to access it regularly, which creates a whole another sort of situation in terms of what to do with this data. You all also know when you acquire this and you finish doing your deconvolution and other things, this data just grows so that you're looking about any 2-week set of experiments needing about 15 terabytes of data here. So what we've done really, and this is all information you guys have all seen, we've built a local area fiber network that is dark fiber, as they call it. So it's 100 gigabit ready, although right now we only use it about 40 gigabits per second. And this allows us to do, and we also created a private cloud that allows us to tackle this information, all this data. We do hold on to the data while the analysis is going on and let our investigators remotely access this and analyze it through use of various different image analysis tools. But at some point, we need a more permanent solution that I think we're all tackling on. I should add that when I said problem-based research, that when we work with investigators, they're not being treated as just a user. We don't just train them and tell them, okay, go with it. We do what we refer to as the collaborative research model. This is where our currency, if you will, is papers and grants and not just money. And that's the approach we have taken in terms of allowing users across from anywhere to come and use. And we've got folks from different parts of Tennessee and around regional states so far come to us and work with Brian and myself and others in my team, both graduate students and postdocs, to work together to collect the data and learn more about what's going on. So where are we at right now? We have something on the order of about 8 different microscopes that do a variety of different things, both labeled and label-free. We work and make these instruments essentially available to anyone interested, sort of in that collaborative research model that I mentioned, where we work with them together to brainstorm what we need, which is the right instrument needed. Sometimes, of course, light sheet is not the answer. Often we found actually that doing some of these label-free approaches, such as nonlinear Raman, is much more powerful, which few people, definitely few biologists, are quite aware of what the power of what it can do. So this allows us to work with them and really think about learning what's going on, not just at the molecular level, but down to the biochemical level to get a fuller picture, if you will, of what to do. These are clearly none of this, certainly not Mantis or any of these instruments are clearly not turnkey instruments, which is why this model of Biomid works better. We actually started at Biomid with the idea that our discovery instrument would sit at Scissor at our core so that users could work with that over there with that 4-fee model while we continue to innovate in that innovation unit or this instrument. However, we found, and it still sits in Scissor, we have one lattice light sheet and one SOPI over there. However, what we found is that it's really difficult in the core model for people to work at the level of handholding and working together that's needed with investigators. And so we often find investigators will just come instead to Biomid instead of Scissor and just sit down with us, brainstorm and work together. And we've been successful in getting our investigators that we work with, getting multiple R01s and other types of grants with the data we've been able to acquire as part of their preliminary research. I should add a couple of new things that are on the horizon, and then I'll wrap up. So there's plenty of time for questions. One is that we have a new facility that is under construction. It was supposed to be ready in January, but now we're told it'll be May, as all construction projects go, where all these microscopes will be housed. there, along with housing facilities and remote access, depending on when we get there. And I'll mention that in just a second. We also are in the planning phases of creating what we're calling the Vanderbilt Visitor Imaging Program, where researchers across any institution can work with us, come here, spend a week at a time, and acquire data, ultimately, potentially, use it remotely as well. With that, I will wrap up because I did not track how long I was going to take for this presentation. Thank my funding sources, thank all the people at the VPC, along with some close collaborators for the projects, such as the preterm birth I've just mentioned, and I'm happy to take any questions.
Wonderful. That was a great talk. Any questions? Either online or in the audience? I need help if it's in the audience.
Thank you. That was a great talk. I was curious about the mechanism by which you are activating the action potentials with the thermal imaging. So did you mention that when you send in a pulse, it creates a little bit of a break in the lipid? I see. Got it. Thank you.
Sorry, was there a question in that?
Yeah, I was trying to understand the mechanism, and I now understand that the lipid bilayer.
So the lipid bilayer membrane just gets loosened, but the isomerization causes them from being this way to go looser, which allows water transport, which leads to calcium influx and so on. So,
thank you.
Maybe I'll pop a question, Anita. Wonderful talk. I love that model that you had. The question is, how do you finance this?
Good question, and we have a short-term plan and a long-term plan. So our short-term plan has been to get some sort of an imaging scientist, if you will, that along with Brian, who will work with these investigators that I am currently supporting from other funds, because some of these projects I showed you data are my R1s that I can help for support. Longer term, what we're getting is, as investigators get some data and are submitting grants, we're asking them to put effort for Brian as a consultant, as well as the imaging scientist, along with a pot of money for any modifications we have to do with the sample arm and so on. And as they're getting it, we have at least 3 of them who've already gotten these grants. We are looking to essentially, this imaging scientist will have their effort covered by lots of little pots of money.
Okay, thanks a bunch. Best of luck. I love that idea.
And so we also are, talking to others. We submitted an NIH, center type grant. They call these BTDDs, biomedical technology dissemination, and I forgot the other D, discovery and dissemination or something along those lines. And didn't get funded right away, but plenty of good comments, and we hope to be able to get some center grants. And if you can get one of those are good for 15 years. So by which time I'll be retired and it'll be some Brian's problem. So, and folks like CZI, of course, as well, are entities too.
So this is Kevin here. I have a question that I think is for all the speakers, but since you won't be at the later session, I wanted to ask your opinion. One of the challenges in academics when you do optics is how to recruit and retain talented staff. That the university regimen doesn't really lend itself well to hiring optical engineers. What kind of challenges do you face there in terms of getting good staff, in your endeavors, and how do you retain them?
Yeah. So, we've got a couple of key people who've grown roots here, let's just say. And so we've been able to keep them just by giving they really just want to do exciting new things. That's what they're luckily not in the business for money, but they just want to do exciting new things. So that's worked. We tend to also work with a lot of postdocs who come for 2-3 years at a time. That's worked. So historically, I have not hired a lot of staff. I am slowly transitioning to getting a couple more staff members as we start this visitor imaging program. But it's a hybrid between PhD students who just are hungry to learn more and do new things, postdocs, and staff. And you're right, we hired somebody who was really good at it, and then Nikon offered him about 3 times the amount of money that I can, and he was gone, right? That's just the reality of academia that we live in. So what I try to attract are people who are excited by the creativity and innovation of what we do, and want the freedom to be able to do that without being constrained by corporate America. And that's where we've been successful in the few that we've been able to retain. It's a challenge, though, huge challenge. Completely agree.
Okay, we'll probably should end there for the next speaker, but thanks so much, Nina.
Thank you. Thank you so much. And I'll try to stick around for the end Q&A, depending on class, because I do have to leave at noon for my class. So
Actually, you do have. Yes, now it's a break, right, Anita? Is that right, Heather and Nikki? Another question?
I think Carolina's asking, but we can't hear you.
Yeah, can you hear us?
Sorry, the room was muted. We have some questions in the room. Hopefully you can hear me now, and Katerina
Oh, okay.
has a.
Yeah, we couldn't hear you. Okay, great.
Thank you all.
Okay, hi.
There's more questions. Sorry, Nina.
Can you hear me?
yes,
Okay.
you can.
Okay. So, hi, this is Katerina from UMass Medical School. I recently joined the ABRF core and rigor and reproducibility committee, and last year they released a great paper entitled Addressing the Environmental Impact of Science Through More Rigor and Reproducible and Sustainable Conduct of Research. And one of the points they were making is that sometimes there has to be some kind of a culture shift toward more robust and maybe less, I mean, you guys are developing some amazing new technology, but sometimes experiment can be done by microscope that may be a little less advanced and maybe cost less, and by doing the things that you're doing, so to build them in-house and in a way that it can be reused and recycled, the parts can be recycled, it can be much more sustainable. And this actually has an impact on the environment and on everything else, and on efficient use of resources. I'm wondering if you have any ideas of how we could start shifting the culture toward maybe less glitzy new microscopes toward maybe more sustainable microscopes, and then we could have more efficient use of resources toward the building of actually new technology that everybody needs.
Can I answer that? I guess I can start off, and then maybe Kevin, you can take over. So that's part of what we are trying, right? By making others just come and use these rather than just creating a whole bunch of these instruments in individual labs. And that's why one of the other things we are just starting to do is also just remote access of these instruments. So you don't even have to have you can save on the carbon footprint as well. I mean, you may have to come the first time to learn and know what's possible, but remotely not only acquire the image, but and analyze the image. And that's where we are going towards, in that sustainable model that you're talking about, especially when you start working at low resource institutions that can't really afford to travel extensively for this data. They're great scientists and they have great ideas and they want to do things, but they don't have access to these things, and they don't necessarily have the funds to do it either, all the technical know-how. And that's why these kinds of models, I think, allow that, both in terms of the environment and the carbon footprint, but also in terms of not just, this also gives us the freedom to keep building new things, because we can build one or two of them and make it available to everybody. So everybody doesn't have to keep building, that I don't have to go lab to lab trying to build. So that's at least how I'm looking at it, sort of from the twofold perspective of how to keep it sustainable. And we do a lot of robust things, of course, to make sure it's reproducible as well. And this is where having two instruments allows you to make sure you're getting the same answer. If you use, different instruments, then you're doing it correctly.
Hi, this is Alison North from Rockefeller University. My question was really for Anita, and I may have missed this. I'm sorry, it's hard to hear in here, but we've had a lot of discussion about promoting career paths for people in core facilities and retaining good staff in standard cores as well. And so I'm very passionate about the standard core staff also having access to new advanced equipment, because I think that's what keeps them motivated. I think it's very important not to separate them into like the boring core with traditional stuff and the exciting new core. So did you say the new facility is going to incorporate it all in one space? Is that how it's going to work?
Yes and no, because no institution is able to make a facility big enough to bring everything together and co-localize completely. To some extent, yes. That's what we tried, which is where we took our first 2 instruments and we put it in our user facility. But it's difficult for them to invest in this kind of model, the traditional core of working with users extensively. They are more used to training people and then letting them loose on the instrument and just being available for advice. And many of the staff there have a difficult time. They know how to use instruments. They don't know how they work per se, which is the biggest challenge we ran. And these are instruments that are finicky. I mean, they're in an instrument that require a little bit of hand-holding. And so what we found kept happening is when users would come and try to use those instruments at the facilities there, anything goes wrong, they have to come running to us because they can't just call a rep, of a company and say, hey, come fix my instrument. And so it's been a little difficult to have both a fee-based core as well as what I'm calling a collaborative, the collaborative model in a single enterprise at the moment. But we hope that when we go to the new facility, scissor instruments will be relatively close. Well, there'll still be a different facility, that they'll be close enough that at least will be sort of kind of co-localized. At least that's the model we're slowly trying to go towards.
But will you be charging for scissor and not for the other core, or will you be charging for both?
So the instruments I'm talking about under Biomid are not charged. When they are in the scissor is purely charge-based model. Their staff is supported through the charge-based model and are completely the charge-based model. So that's why we tried the transition space, and it's just, I found it just difficult. And so the scissor operates on a charge-based model, but we operate on a publications and grants-based model. Our charges are publications and grants, if you will.
Okay, thank you.
Okay, thank you. That was a great session. Everybody did a marvelous job of staying on time. Thank you, Kevin. Thank you, everybody. We're going to take a short break, 15 minutes. We'll be back at 45 minutes past the hour, and then we will wrap up. So thank you very much to everybody that presented this morning. If you're able to stay on for the Q&A afterwards, we would really appreciate it, but we also understand you have other commitments. So, thank you.
Thanks so much, everybody.
Okay, Kevin, I think probably we'll hand off and I'll let you do introductions.
People
are wandering in, but we'll let you take it away.
All right, thanks everybody for coming back. Our next speaker is Shalin Mehta from CZI, the Zuckerberg Biohub, and he's a leader in computational microscopy, including some field-leading work in polarization imaging. Thank you so much, Shalin, for giving the talk.
Thank you so much for having me. I really enjoyed learning about Abhishek's, Reto's, and Anita's work and their efforts at deploying. So I'm going to talk about our efforts at developing and deploying computational imaging methods, where the computation is an integral part of the acquisition in the form of calibration and in the form of reconstruction. And I'll primarily focus on label-free imaging methods. We heard the promise of label-free imaging from Anita, and the kinds of methods I'll talk about, they're slightly different. They rely on the single scattering, single photon interactions rather than nonlinear interactions. So as a start, I would like to give a bit of an overview of our set of projects and how we think about imaging and analyzing the dynamical systems that cells and tissues are. So the first task is to encode the dynamics, and we utilize different single photon interactions to encode the dynamics. So in this movie, we are looking at a dividing cell, as you can see, and this movie is acquired without any labels. And what we have done here is to encode the density and the orientation of the biological material using phase and polarization of light, and reconstructed those physical properties of the material, and then color coded that. in an RGB lookup within a specific RGB lookup table. So this movie illustrates that one can encode many components of the biological architecture of the cell, in this case organelles, without having to stain them. And this is a way to multiplex visualization of many structures and their interactions. Now, not all the structures that are of interest and all the dynamics that are of interest are encodable with without label, and so it is really valuable to do correlative imaging where you use fluorescence to look at the signaling molecules, to look at the endomembrane system structures that are very faint when looked at without label. And so we developed these methods where we combine label-free imaging and fluorescence imaging, and do this at scale, do this in 9612 plates across whole embryos and so on. Now, all of this in microscopes result in a large amount of data, and typically the first step in reducing these images into biological insights is to find the components in the system. And we develop segmentation methods. They're needed primarily for label-free data sets. There are well-established and robust segmentation methods available for fluorescence modalities. So we have been developing robust approaches for translating the label-free images into images that look like fluorescence. So we call this a virtual staining approach. And once the data looks like fluorescence, some of the more classical segmentation methods can be applied to find these components. We also leverage the inherent dynamics that are encoded in the label-free images to learn the architectural states. So for the 20 minutes I have today, I'll just focus on the top half and our efforts at building out these methods, as well as making them first useful within our institute, making them useful at scale within institute, as well as enabling their adoption in other labs where there is where they can be valuable and useful. So as a background, the field of computational imaging is concerned with solving this inverse problem, which is to measure the unknown architecture of the biological sample. So let's imagine a cell, and let's call that what we would like to know is the global architecture of the cell and maybe distribution of some organelle like a mitochondria. And the forward problem of imaging is to encode this information in intensity. And we do that, all of us do that with a microscope. We send in some light, that light interacts with the material. The material may have a label, and then it will emit a photon, or it will scatter a photon. And one can, the design goal with this step of imaging is to encode the invisible biological structure into visible images. Now, that process is mathematically not super complex. You can model that, but there's always also noise that gets added. And then, we get images like this. So this is a label-free image of a cell, and this is the fluorescence image. Now, these images are pure visualizations. They are not necessarily representations of the architecture. They're poorer representations of the architecture. So once we have this mathematical model, it's actually interesting to think about how we can get to a better estimate of the biological architecture. And that problem is an inverse problem, given the images, which is this measurement y, which encodes the unknown x through some operators, and it suffers from noise. How do we estimate the sample? It's an interesting question, and doing so is valuable, because Now we get closer to the biological architecture. In some sense, by solving this inverse problem, what we are trying to do is to remove the microscope from the, remove the effects of the microscope from the data to the extent it is possible. Microscope is a filter, it's a lossy instrument, so we can't undo all of the distortions that are caused by the imaging system, but we can undo several and make the data more useful, more interpretable, more analyzable. So that's the approach we take. Now, this approach has also been applied across many different scales. I wanted to show this data sets, these images from about 30, 40, 50 years ago across different spatial scales. So this is an image of a bacteriophage. This is an image of a immune cell chasing a bacterium. This is one of the first movies of a dividing spindle. And this is an MRI image. Now, what's interesting is that across all of the scales, the information about the architecture is encoded through some physical properties of the structure. Here we are looking at the density of shell electrons. Here at the visible range, in the visible range, we are looking at the density of the bound electrons. Here we are looking at the density of protons using magnetic fields. What has happened over the last few decades is that computation, the ability to model this image formation and the computation has completely transformed the amount and the quality of information we can extract at all of the scales. So cryo-EM relies on aligning different particles and then really getting to the structure. And now a lot of structural biology happens with cryo-EM. At the visible wavelength, now you can encode a lot of information, both density and the orientation at the same time and recover that efficiently. These methods work well, so you can apply them at scales and look at the entire tissue. At the scale of organs, now one can map the connectivity in the brain tissue by measuring the flow of water, by measuring the diffusion of water. Now, it's worth pointing out that the color here does not represent the fluorescence channels. It represents a different aspect of the biological architecture. It represents the orientation of the architecture. So I want to say a few words about that. So I really like this phrase from Rob Phillips, that the biological architecture is tensorial. What this phrase implies is that when we think about the structure of a cell, there is not just a positional arrangement or a positional order, but also there is an orientational order. In other words, when we think about an axon, the axon exists not just in a specific location, but it also has a specific orientation in a 3D space. And representing this aspect of the biological architecture requires quantities that are not just scalar, not just vector, but slightly higher dimensional. In other words, tensor quantities. So if we represent the axons in terms of the diffusion of water in brain, then we are going to need a few parameters. We are going to need directions in which the water flows and the magnitude of the water diffusion along those principal directions. So this is in some sense higher dimensional quantity than a vector, but it's elegantly representable with tensor, with matrices, and one can exert a lot of computational control over them by using this representation. And the data that I'm going to show today, it is the primary source of information is another type of tensor. It's the refractive index tensor, or more precisely speaking, permittivity tensor. So the idea here is that when we have biological structures such as a lipid bilayer, the refractive index is a function of the orientation of the lipid bilayer. We have slightly higher index perpendicular to the plane of lipid bilayer, and we have a lower index in the plane of lipid bilayer. So the refractive index or permittivity tensor looks like an ellipsoid. And the methods that I'll discuss, they all encode these permittivity tensor with different completeness, different resolution, etc., etc. So this is a video from Lachlan Whitehead, who makes this fantastic animations, which illustrate how the refractive index tensor or permittivity tensor can be encoded in intensities. So when we send in some light, and the light interacts with a molecule, it induces electric field inside the molecule, or induces a dipole. And if we have an arrangement in space and orientation of these molecules, these dipole distributions also have a spatial and angular arrangement, and they, you get induced dipoles like this. If you have a shell of lipid bilayer, imagine an axon. And now this movie, the movie you saw was indicating temporal dynamics that were very fast, but over this time we make the measurement, we are recording the effects of the net radiation. And the net effect is that the materials can be represented as this ellipsoid, whose isotropic component reports the density. The anisotropic component reports the orientation, and the orientation is a vector quantity, so it has a magnitude, and also the 3D orientation. So that's kind of the big picture link between the architecture and the kinds of measurements that one can make with label-free methods of all sorts. So we are thinking of the biological architecture as this collection of cigars. That's in this case, lipid bilayer can be thought of as a collection of cigars, and one can interpret a lot of aspects of the symmetry from this data. So I'm going to now take some simple examples here of how the encoding works in real experiment. So let's imagine that I have some glass bead immersed in a water. It happens that this glass bead is going to be transparent if it is a small glass bead when the microscope, when it's in the focus of the microscope. When it goes out of focus, when the microscope's focus changes, the bead will appear bright or dark, depending on the direction in which you defocus. So this brightness and darkness, this rate of change of intensity encodes for the density of the bead and the size of the bead, etc. And if you have another situation where this bead is replaced by an air bubble, then the contrast variation, the direction of contrast variation reverses. And one can write this, one can express this as equations and invert that. So now in a real cell, when you do that, when this is an image of a real cell where you can see that this lipid droplets look bright on one side of the focus and dark on the other side of the focus. When you model that and you invert this contrast variation, you get sharper images, but also at the same time, you get more quantitative representation of the architecture. And when we encode the orientation, we use another property of light called polarization, which interacts with the material, and the polarization changes. This is what the polarized light images look like, and one can apply computation to recover the alignment and the orientation. So that's the big picture approach here. This is how the methods work. Now I'm going to show some images of cells and embryos at different scales. Our focus at the Biohub is on understanding the mechanisms of infection and enabling drug screens in the context of infection, and using embryos, using zebrafish embryos as models for development and as models for immune system dynamics. So the images I'll show you, they are drawn from these domains of application. We are looking at here a dividing cell, and on the left, we are looking at the phase image, and on the right, the orientation image. The phase image shows the variations in the density of the cell, and therefore it shows the boundaries of the cell. You can see condensation of chromosomes, lipid droplets, nucleoli, etc. So you can see several structures here. Now the orientation image, in the orientation image, the distinct structures are membrane and membrane-bound organelles, the plasma membrane, as well as the spindle, which forms the liquid crystalline structure. And this is an extension of work that I started with Rudolf Oldenburg while I was at MBL. And when we go to another scale, when we look at a developing embryo, this is the kind of data we get. So this is the standard bright field movie that you would get. Now, when we use polarization of light, we can see the tissue flow with higher contrast. But at the same time, we can begin to discern the mechanical strain as the embryo develops. And it's also intriguing to see these patterns of oriented structure as the embryo, as the tissue undergoes morphogenesis. We apply this approach across entire zebrafish too. We are looking at immune dynamics across the zebrafish, and the approach here is to look at the tissue without label. So here we are looking at the gut, for example. In the density image, the somites from polarization image, you can also see blood flow, and look at the immune cells themselves with the fluorescence. And here we are looking at the immune cells in the context of the tissue with fluorescence. This is yet another example applied to imaging of the myelination and the connectivity in a mouse brain tissue. We are looking at a coronal section of mouse brain, and this is a stitch of, and you can see some stitching artifacts. This is a stitch of many fields of view, but this data collection took less than 2 hours, and it's very high resolution. So the primary source of signal here happens to be aligned lipids in the myelin, and when we zoom in into that, into the corpus callosum, a part of the tissue, we can appreciate that there's a lot of information here. We are able to resolve in 3D the cross sections of single exons, so there's a lot of information here. Now, so this approach, these approaches have been now, we have developed them for use at the Biohub, and we are using them routinely at the Biohub. And we have explored some design space. We have tried to make try to develop a version that is very sensitive, a version that is very fast, a version that has the highest resolution in 3D, and also a version that is gentle. And in this version, we are combining the oblique plane microscopy that Reto talked about with the very fast label-free imaging system. And we also call it Mantis, coincidentally. Okay, so this is kind of the survey of the methods, and an important thing to point out here is that these label-free methods that use single photon interactions can be quite straightforward to implement and quite inexpensive to implement. So this is a build video of the microscope that led to the 3D fly-throughs I showed through the mouse brain section. And you start with a standard microscope. It can be a wide field, it can be confocal, it can be an OPM, and then you add a path. And that path typically has a detector, and in this case, machine vision detectors work really well. And you add some way of controlling the patterns of illumination, so angle of illumination and polarization of illumination, and then you're done. So the complexity of the hardware is not that high. One can build such a light path in an afternoon and get it aligned in another afternoon. The complexity resides really in the algorithms and in the software. So here's kind of an illustration of how the inverse algorithm works for the microscope that you just saw. Let's imagine a distribution of exons, some in plane, some going out of plane. The microscope encodes this distribution of exons in these images. And the inverse algorithm uses a set of optical transfer functions. And the problem is somewhat similar to structured illumination that you heard about today morning. And you then apply the inverse algorithm and recover the map of density, alignment, symmetry, orientation, and tilt. So how do we make this accessible? I think that's a challenge we have been working on. One important point to make here is that there are some parameters that need to be tuned as in these algorithms. And for inverse algorithm, a parameter is regularization, which is an approach to deal with the noise. And as you tune the regularization parameter, you will get different reconstructions, and some reconstructions are right, and some reconstructions are obviously not right. So we are thinking about how do we make this choice of parameters either automated, or if a human intervention is required, how do we allow a human, provide guidance to choose the right parameters so that the downstream biological analysis is reliable, accurate, and consistent. So our deployment effort looks like this. We think of how to deploy computational imaging methods in the form of an n-dimensional image I/O stack. And it starts with state-of-the-art microscopes, the hardware. The layer above is microscope automation. The layer above that is n-dimensional image I/O and visualization, and the plugins for controlling all of this, and finally doing machine learning on top of it. So these are some of the repositories that we build on the open source tools, and we are contributing back to the open source tools. These are some of the repositories that may be of interest to the community here. Biohub is a repository for converting the TIFF stacks into a ZAR store, which is a format that allows better organization of n-dimensional data and better read and write performance. This is broadly useful, and the community is moving towards ZAR store, and open microscopy environment now has a metadata standard around it. And we are contributing to that. We are using that and contributing to that. Our algorithms are all packaged in a repository we call WaveOrder. It provides simulation and reconstruction algorithms for a variety of methods. And we try to make them easy to use for engineers at this point, where we have clean API and clean CLI. And for the end user, for the biologist, we are packaging these algorithms in the form of workflow. We currently rely on Napari to be the n-dimensional visualization engine, and we build this Napari plugin, Napari plugins for making our pipelines accessible. The pipeline that enables the label-free imaging method is called Recorder. As I mentioned, it builds on all of these components. We have taken this to AQLM in Woods Hole and learned a few lessons. And I think I want to add here on some of these lessons. What we have found is that in our work, we have been able to package these algorithms reasonably well, make them accessible via CLI and API. A reasonably experienced microscope builder and programmer can use these tools. We are currently in the process of solving the integration with acquisition engines and streamlining the user interface. I'm really open to discussion about how to pursue this, right? How folks are thinking about this, and what are different approaches to now take these tools from the hands of engineers and really make them very easy to use for the biologist. And I think it's going to be a process, and it will require all of our energy. So with that, I want to close and thank the amazing team that does all the work and that collaborate on these algorithms and on this repository. Shalin is driving Recorder and WaveOrder. Ziven is driving Biohub and is contributing to both of them. And Ivan develops the automation pipelines that we use at the Biohub. And I hope I have left some time for questions.
We're going to save questions to the break at the end. So we have time there, so we can go to the next speaker. So if you're able, Shalin, to wait to then, that'd be great. So we'll do our next speaker, but thanks so much, Shalin. And so our next speaker is Alenka Lovy, who I think is in the audience there. Welcome, Alenka. She's going to be talking about her efforts to bring the Flamingo light sheet to South America.
Hey, hi. You heard from me a little earlier if you were present at Labby. I'm sorry if some of my slides are duplicate material, but I'll be focusing on bringing Flamingo light sheets to South America. So it'll be a slightly different. Thank you so much to the organizers for inviting me. I've had a wonderful time interacting with all of you. It's been a really nice experience to be here. Just a little background on me. I moved to Santiago, Chile 4 years ago. Before then, I was working at Tufts in Boston, Tufts School of Medicine, and I managed an imaging facility there for 13 years. And I kept trying to start my research program, and I found it impossible to run an imaging facility and have a research program. So when I moved to Chile, I was like, okay, I'll focus on doing research. And as you see, I'm gravitating towards microscopes, and I'm always working with microscopes. So our microscopy facility got established in 2016, along with our research facilities. So research is quite new at our university. And I just wanted to say we have a confocal microscope, an epifluorescence microscope, a cytometer. We have Imaris software that some researchers bought together. And importantly, we have Ramon Ramirez, who manages all the equipment and trains researchers on the use. So we do not charge fees, and we're too little. We support our university for now. We have some centers of research and with accredited PhD programs, so students are coming in to use the microscopes. But that's all changing because we were able to acquire the light sheet microscope with this FundaKeep grant, which is the National Instrumentation Grant. And so these grants support new cutting edge equipment. And basically it's the only way to get new equipment. And you cannot use this mechanism to acquire similar equipment. So even if you can justify it, it's a no-go. So, and the university has to contribute a certain percentage, and then the agency pays the rest. And so the requirement is that you share this equipment with the entire region. And it requires hiring of a technician to operate it. And just to review, I mean, you've heard so much about light sheet fluorescence today, but as you saw, it enables 3D imaging of live and preserved organisms with minimal phototoxic and photo-bleaching effects. And it's really fast and can image a very long time. I had a slide here. I think I have my old presentation, but that's okay. Because Reto told you how light sheet fluorescence works. So basically, you have a sheet of light, a whole XY plane that you're using to scan your specimen instead of like in a confocal where you scan point by point, right? So that's why you don't have that cone of bleaching throughout your whole specimen. And that's why you have less bleaching overall, and you can image super fast. So when we saw this Chan Zuckerberg initiative opportunity to expand global access to bioimaging, we were super excited to apply, and even more excited to be funded to build a light sheet bioimaging hub. And with this, we were able to establish, with this funding, we were able to establish Lithium, or light sheet imaging at Universidad Mayor, and we're able to organize conferences, workshops, and seminars to disseminate the technology. And we're also opening our facility in a remote fashion where you can send your samples, we'll pay for the shipping, and we'll image your sample with you online to see whether this is the right technique for you. We were able to hire Anibal Vargas, a postdoctoral researcher supported by CZI, to help us in all these light sheet events, and also to sustain future light sheet imaging he's going to learn how to construct more affordable light sheet microscopes so that if we have a greater demand of light sheet, we can kind of aid in constructing new ones. So we were quite worried having the only light sheet in such a, well, on the continent. How are we going to support all the demand? And so we started talking with Zeiss and Microx Chile, which is the representative of Zeiss in Chile. And they were very supportive, and they said, if you have a very high demand, we can arrange to lease you some instruments for 2 or 3 months at a time. And so with a little bit of organization, we were calm that we had a solution. And then at the time I was attending the MBL Light Sheet Conference, and I saw Jan Wisken's Flamingo project, where, I mean, I guess Jan has been involved in developing light sheets for a long time, along with Michael Weber. But this Flamingo project was amazing because you can pack it up in a car and take it to another lab. And in fact, that is the Flamingo project, the concept of the microscope coming to you rather than taking your precious specimens over to the microscope. So I thought this would be really great in Chile, right? So I contacted Jan, and he was super supportive, and he said, yes, he would send us a Flamingo. And so great, we started looking into it, and we were, I guess there's a temporary admission into the country, so you can borrow the microscope, but we had to come up with an insurance policy that was around $20,000. And if you got delayed in keeping the microscope longer than 120 days, you had to pay the full value of the microscope. And then there were a whole bunch of mysterious papers that we needed. And I started hearing stories where people imported stuff and it got stuck in customs, and then they couldn't get it back. And I felt super bad to borrow Jan's microscope and get it stuck in Chile and not be able to return it. So basically, out of fear, I decided to stick to learning how to build these microscopes. So Anibal is actually going to Germany tomorrow, and he'll start documenting the process. He'll stay there for a month and work with Michael Weber and Jan Wisken and all the members of the laboratory. And in the meantime, if we have a lot of interest, we were thinking to gather researchers to write another Fond d'équipe grant for a, traveling flamingo, hoping that it was a different enough of a concept that they would accept our application. And actually, I was sharing these ideas at the CZI annual meetings, and I ran into a lot of people that were very interested in bringing light sheet to their countries. And luckily, Jan and Michael were very supportive. And so when we saw another call from CZI to advance imaging through collaborative projects, we were super excited to come together and try to make this happen. So we have some members from Chile, like Leonardo Valdivia, Felipe Kurt, and Cesar Cardenas. And from Brazil, we have Gregory Kitten, Christopher Kuchmarek, and Vinicius Rivas. And from Uruguay, Leonel Malacrida and Andres Camaid. So we have a nice mix of scientists and developers. So hopefully we can pull this off. So we plan to build 3 variations of Flamingo microscopes in Germany and distribute them back into Brazil, Uruguay, and Chile. And each one will come with its own configuration, but I think because these systems are modular, we're gonna have the capability to switch between configurations, which I think is really great. And the idea is we'll have workshops between the facilities where the flamingos are placed, and we'll award pilot grants to promote light sheet techniques. And then eventually we'll organize a conference. But the idea is that the microscopes can travel to various places within the country without having to cross the borders and deal with all the bureaucracy that's involved with that. And we've identified many researchers that expressed interest in using the microscope. So we're super excited. And we're looking to construct them in Germany and donate them back to our countries, because in this way we can maintain the affordability, because as you know, by getting parts to South America is really expensive. And when we did the calculations to buy all the parts that we needed, it at least doubled the cost of the instrument. So we're going to try to construct them in Germany and donate them back. This is some of the paperwork that's necessary for that. And we're starting the process of learning how to accept a donation. It has to be through the institution, and you have to work with customs officers, and they have to approve how you're going to accept the instrument and what you're going to use it for. But this is all stuff that I hope to be able to tell you about later once I go through it. So in the meantime, we will be growing our light sheet network of researchers. So Jan has been super nice to present informational webinars on his Flamingo system, and then take us to his lab and have an interactive session and ask him questions about, how all the nitty-gritty details of how these microscopes work. We have recordings of all of our webinars on our website at lightsheetchile.cl if you're interested. So some of the other workshops we've been organizing deal with sample preparation, because that's actually the obstacle to getting people to come try this new equipment. People that are at the confocal need to, re-embed their samples in agarose, because the light sheet is like a microscope that's laying on its side, and you have to sort of hang your sample inside a chamber. The two objectives on the sides are making the light sheet, and the one in the back is laying on its side, and you have to dangle your sample immersed in water usually, or buffer. And so I think that's the biggest obstacle to getting people to come, but it's actually not so bad to prepare the sample. It's just different. But so we're holding a lot of workshops on that, and helping people one-on-one to prepare their samples. And Ramon Ramirez is also helping people do clarity. He has a nice clarity set up, so people are welcome to come, clarify their samples, and then we make appointments to image them. So our biggest workshop was the expansion microscopy workshop that we ran, and I guess that was a week long, and it was international. And this is what taught me so much about how to actually put on a workshop. And so Ulrich Kubitschek and Juan Eduardo Rodriguez came from the Clausius Institute for Physical and Theoretical Chemistry in Germany. And we invited them, and they basically put on this wonderful workshop where they taught imaging fundamentals, and then got into light sheet microscopy. And Juan Eduardo held a laboratory session where they expanded everybody's samples. We had a test sample that we worked with, but then people brought their own, because of course people always want to see what their own samples look like. And then we arranged imaging on wide field microscopes, confocals, and light sheet to compare the technologies. And then we had Mauricio Cerda and Victor Castaneda from University of Chile come and work on imaging processing and teach about processing fundamentals. So just to show you, I mean, maybe many of you have organized workshops, but this was my first one, and it was really a huge learning experience. So because we had international participants, we had to find hotels and bus coordination. And I thought, okay, well, the university has buses, maybe they'll let us use theirs. And they did, but everything was for a fee, and everything was like a lot of documentation to arrange. So I was very surprised that even at your own institution, you have to do so much paperwork. But that was fine. We got hopefully a little discount on the hotel from the university. And then we had to arrange a huge auditorium because we wanted to do the workshop as hybrid and virtual, and we wanted to record it and make it accessible to as many people as possible. But that worked out well. The laboratory was beautiful, but we didn't have an internet signal there, so we weren't able to share the laboratory experience with everybody else. But I guess that's kind of difficult anyways. But getting the supplies, believe it or not, it took 7 months for us to get our supplies. So I was very happy that I prepare a very long time ahead of time. But yeah, if you forget something, I think you just have to make do without it. And, you know, occupying the microscopes for a week was a little tricky. They're small rooms, so we had to kind of split up the groups of 30, and we had 30 people, so it was kind of hard to rotate through all the microscopes and have time to see everybody's samples. But it worked out. And then finding computers that were able to support the image processing was also tricky. So luckily we found a graduate computer room that was able to support Imaris and Huygens. And it was really great to get support from these companies who gave us free licenses for the week to use for teaching purposes. And then, of course, because we're on a kind of isolated campus, we had to arrange for the food. And luckily, there were some señoras that usually come and provide food, home-cooked food for the students. And we made an agreement with them because this was a time when there were no students around. So they agreed to come and make us lunch for a week. So that was really good. And then, so it was very valuable to hear how the students reported on their experience and their results. And I think overall, we had a very successful workshop. And I was really, really happy to go through that experience. But I really under-budgeted for this, and I didn't know, but if you have an international workshop, you need $10,000. Like, I wouldn't ever try to do it for less. And I tried to, you know, get the best deal on everything, and still, I hardly made it for less than $10,000. And I found it very difficult to access the grant money quickly enough to actually make things happen. So I was feeling very lucky that I had some personal savings so I could pay for the whole workshop and then get reimbursed later. But I would really appreciate more institutional support here because, and then afterwards, I looked like that, just buried in papers trying to get all the reimbursements. But I think, you know, trying to do research and dealing with that, it's almost a full-time job. So it was a surprise to me. But on another note, I think there's more sustainable ways to run workshops. I mean, you can get local teachers and you don't always have to cover the food. You know, you can tell people to bring their food, but of course it's really nice to have the food covered. But, you know, as an example, Jacques Paysan came to visit us and he still owed us some training on the light sheet microscope. And so we gathered all the frequent users of the instrument and everyone had like 5 hours with him throughout the week. And so they got to ask him very specific questions. And then we organized a workshop where everyone presented their data and their findings. And then we brought in new people to try the instrument. But just to bring it back to Flamingo light sheet microscopes, when we travel out to courses, it will be so much easier to bring the Flamingo with us. So, for example, this EMBO developmental biology course in Quintay, which is 2 hours from Santiago, we were super lucky to have the support of Zeiss and Microx Chile, and they arranged to send a whole light sheet system over there, which was also in itself like $10,000 just to get the system out there, and then to have the teams assemble it, disassemble it, and run it throughout the course. It was a really big effort, but I can imagine the Flamingo would really be very efficient in this case. But it was really fun to be able to image all the different embryos and the students would stay up till 3 or 4 in the morning imaging. It was amazing. And then, of course, traveling with the Flamingo to other courses like the Small Brains and Big Ideas, another EMBO Global Exchange lecture course, and to the annual Chilean Society for Cell Biology, I think will be great. So we really look forward to constructing these microscopes. And I want to thank the organizers again for this wonderful meeting, and to all the light sheet researchers and collaborations that make this project possible. Let's keep building our network. Please, if you want to get involved in light sheet in South America, please reach out. You can sign up on our website at lightsheetchile.cl. And thank you very much.
Wonderful, thank you. Kevin and everybody that's still with us virtually, we're going to move into a general Q&A. So those that presented here, Abhishek and Alenka, if you would like to come sit at the front, then Vanessa can light you. And I'll hand it over to you, Kevin, for the Q&A. But, you know, drop something in the chat or anything if you need help.
Great. Any questions to start, or I can ask one? Any questions in the audience?
Here in the audience. So I'm Adan Guerrero. So thank you for these lovely presentations. So this is a question more for the organizations that provide support for these initiatives, for these educational initiatives. And it's about having the problems that Alenka just stressed, that organizers normally and commonly and often and all the time have to put personal funds to organize the workshops. Otherwise, that doesn't come to a fruitful end. So I wonder if there is any way to support everywhere in the world to have liquid funding to start, and then after just recover that money. Otherwise, the organizers really face economical issues before the workshop.
I mean, that would be wonderful. I think even if the institution was able to give me a credit card linked to the grant money, that would also be very helpful for a start. But I think having a reserve would be good.
I also organize a light sheet workshop at the MBL, and I also face similar problem because I do have a credit card, but I max out in the first day or two, and then I have to use my personal credit card and get it reimbursed, because even though process is fast, but there is a delay. And when we are hosting workshops, we want some decisions now rather than tomorrow or day after tomorrow. So it's a tricky balance.
Hi, this is Katerina from UMass again. I just have a comment because the question that Adan asked made me think of something. So recently I stumbled upon something called Open Collective and Open Source Collective, which are fiscal sponsors. And so they are actually working for various organizations, and particularly for the open source, to do exactly what you're talking about. They are based now in the US, but then they're working with other countries to do it also in other countries. Basically, they are a place where money can be hosted and used to pay without going through a lot of the paperwork. So the paperwork is extremely streamlined, and they have very low overhead. We are exploring the use of that for example, for supporting some of our work, and I'm happy to work with others to, reach out to them and see what could be done, because it strikes me that it could be a way of solving this.
Do we have any questions? Excuse me, do we have any questions online? If not, could I ask a question of you all? If you had a question you wanted to pose to the audience here, you all generate these wonderful technologies, and you've highlighted some of the things that are drawbacks for you for actually getting them into the hands of the users. Can you sort of, in a nutshell, provide some feedback? And maybe we'll put it to the folks in the virtual audience first of all to, you know, if you had one wish, what would that be? Can we put that perhaps to Reto first?
Sorry, I was.
Sorry, shall I repeat the question? So if
Yes,
we have everybody in the audience here, if you wanted to share with them right now something about what it would do to help you to get your technologies into the hands of users, what would that one thing be?
Yeah, I would for a long time love to hold some sort of workshop for certain technologies, but you know, getting the funding, getting the organization done, it is hard. So yeah, I think I would generally think it would be great having funding mechanisms or opportunities to better disseminate technologies when they're popular, you know, if there is a demand. I often think of how Janelia, spearheaded by Eric Betzig, how they disseminated the lattice light sheet, which was not a simple technology from an optical standpoint, but they were holding workshops, they had instructional videos, so that was a, how to say, kind of, to me, a model how you can disseminate. And on our side, we're lacking far behind. We were both probably direly understaffed and underfunded to you know, tackle something like this. But I would love if there is, for example, interest in this OPM technology to you know, have a sort of workshop or people come together that are interested.
So it sounds like if anybody is interested in hosting a workshop, you found a collaborator here. So please get in touch.
Yeah.
Shalin?
I think making the hardware available via kits would be very enabling. We all develop different modules these days, and a typical biological research project requires integration of some of these modules. And if these modules are available, this cutting edge approaches, new approaches are available as modules via Thor Labs or some other entity, I think that reduces that in some part solves the problem of accessing the hardware. On the software side, we struggle with making our plugins work well on multiple microscopes because of a bit of the fragmentation in the acquisition space, in the acquisition software space. I know there's a lot of momentum. There are a lot of groups developing different acquisition software. If as a community we figure out 2 or 3 platforms that work well for different kinds of methods, then that's great. And then one can build software stack on top of it. So fragmentation in the acquisition software will be great to address. And the getting some of these designs available as kits, that will be I think enabling.
I think dissemination, just word of mouth and passing along the information of, you know, what we're doing, or just to go to our website and sign up and get networked so that we can be in touch about what's happening in Lightsheet is really the way to go for me.
Med.
I agree with all of them. I mean, certainly funding is very crucial. A lot of times we can get funding for instrumentation because we can justify as a research projects, but getting funding to support our staff who can help disseminate these things, that's also very crucial, because without human resource, we can't go very far, even though we can have all these tools and technologies. And the other thing, as Alenka highlighted it very nicely, behind the scene, we have to do a lot of paperwork. I'm not complaining. I'm thankful to my institute. I get a lot of support, but still we end up doing a lot of paperwork. So having some investment in human resource will make this process faster or enjoyable for everyone.
So it's a fascinating session, and we've seen some amazing examples of systems which are on imaging tables, sort of blown up systems, which in a way should be more accessible because you're building from components, you're doing it yourself, you're learning the the real nuts and bolts, literally, of all these systems. And yet it strikes me that a lot of these systems seem to be in extremely, shall we say, high-powered institutions. And so where in theory, they should make the technology more accessible and more available to lower resource environments. I get the impression that it takes an MBL to support Abhishek's work, and it takes, all these great institutions. We're thinking back to what Leung mentioned in his talk. It takes a Howard Hughes Medical Institute at Genelia Farm to support what they're doing, and I think we've got a little bit of a dichotomy there. So and at the same time, something that Leung mentioned, the build it and they will come model. We've seen that there are problems with that model. It would be wonderful to be able to go to the Marine Biological Laboratory. It would be wonderful to go to these great institutions to where we've seen today. But it's very, very hard, and we have an energy barrier and we have a psychological barrier to get over. And I see this as a sort of and as Alenka put out, when you do become very, very brave and try and set up a real sort of high-end sort of build it yourself philosophy, there are a lot of difficulties in getting that set up, and there are a lot of barriers to still be overcome. And so it seems to me that there's a breach here. There's a breach. We all want to do it. We can all see the benefits, but it just seems to be something, an element that's missing, something that's just stopping us bringing those two halves together and making sure that this technology really reaches its maximum impact in areas where we'll have that. So I'm just inviting reflections, really. I mean, is that a reasonable summary, or are we still there? And what is that magic something or some things that we need to get across? And I suppose if we had that answer fully, then we wouldn't be sitting here. But ideas really is what I'm thinking about. So.
I can add one thing. One thing that hasn't come up yet is that there's a major challenge in what everybody's doing, including all the speakers in my work as well, and that there's really no open framework. That there's no GitHub for hardware, right? That when you think about the world of hardware and how to share it and how to know expertise and training and parts, we're all on our own, and there's not a good system, right? And so we all kind of do our own thing, and a lot of it's very hard to maintain. There's a little money for that. But if you look at the open source software world, its ability to disseminate to low resources, it's been way more successful because it has the ability to have a GitHub-like repo and ways supporting it. And hardware is way more challenging than sharing software. Someone who does both, I find it way easier to do open source software than open hardware. So we have been talking to some of our collaborators on Mesospin and other kinds of complex systems, and Yahoo Skid and others. How could we team resources to have some sort of framework for sharing expertise in the parts list, right? And also how you build it. Right?
Anybody to that? Or anybody? Here we go. We have a question from the floor. Okay, go ahead, Shalin. Question from the audience then.
I have a thought to add to Kevin's comment. I agree that we haven't figured out a model for sharing the hardware designs. Maybe there are some lessons to be learned from how the maker community, 3D printing community has developed websites where you can share the designs. But it's going to require injection of people who like to do this kind of things, and you know, that they like to build this community resources.
Can I add one thing? While we are figuring out how to share and disseminate all these things, one thing, I have quite a few international collaborations. So one thing I request my collaborators, if you are coming for a conference or another meeting, then swing by to MBL. And if you come to MBL in non-summer months, housing is much more affordable. And then we can work together and try some pilot experiments. So there are a lot of ways we try to optimize our resources and make sure that it's, as you said, it's accessible and equitable across the board. So there are a lot of challenges, but as we are figuring out, I think we need to keep trying things and see what works best for all of us.
Ready? So I have a question, but actually my question is for Peter O'Toole, who I thought had run away, but he's actually still in the room, thank God. So first of all, I take my hat off to people like Alenka who've organized a whole course, because organizing it at the NBL with all the support we get there is hard enough. So congratulations, that is a tour de force. But I think it really highlights, as you said, the difficulty of organizing courses, and everyone who's doing it and has never done it before, there are all of these things that hit you down the road with surprise. But there is an organization who has been organizing courses forever, and that's the Royal Microscopical Society. And they not only organize courses in the UK, but they organize courses in other countries, and they know exactly what they're doing. So my question for Peter, who is now president of the Royal Microscopical Society, is, do they organize a course for teaching people how to organize a course? Because if not, could that be done virtually, telling people how to set up a course?
I can't see why not. So yes, if there's demand, we can do it. Email me to remind me when I get back to the UK.
Just a comment that there are also local network organizers who like organizing things and have experience with that. And while we can't always handle all of the in-country logistics or institutional logistics, we are here for advice and are happy to help where we can.
Hi, just to follow up on what Chris was saying. I mean, I think I don't have necessarily sharing the information is definitely important, but at the end of the day, we will need people and salaries. And I think it strikes me that we hear a lot about the, creating jobs. I mean, everybody's saying that they want to create jobs. So this seems to be like something really that could be helpful to create jobs, because it would be basically, if you train somebody to start building this kind of microscopes, that could be like really a way of having a job for a while, for the future. So I think we just have to, I think there has to be some advocacy effort to make people realize that these kind of training courses can really generate. So, it's just a question of shifting money from maybe buying enormous, super expensive microscopes versus this kind of microscopes that can be reused, and they may be more efficient in terms of those expenses, and at the same time create jobs.
So to comment, one will come from me, another from Claire Brown. So the first is about how other organizations support their people making courses all around the world. And I can give the example of the company of biologists, in which they offer administrative support for taking care of the financial part of the workshop. So I haven't had some of those support, but I've read the call, and now I know that there are now some initiatives that might happen in Latin America. So we will see if that model might have an important role or it becomes more difficult. So, and regarding organization courses, I'm passing the voice to Claire.
So when we ran our Train the Trainer course last summer, we had a session on course logistics, and we started putting together a booklet, which we want to also include checklists of things to do and when to do them, you know, like booking your venue 6 months to a year ahead. And Adan and I have been talking because we know Yuri is a master at this. And also we thought maybe it would make sense to do it under the global bioimaging umbrella and make it available to the whole community that way. And perhaps we can do it in collaboration with the RMS if they already have documentation and things that are out there. So it could be a real community effort. So I'm going to bring it up at the GBI meeting in Stellenbosch with Leb. So we'll see how that manifests. But I think we were thinking also of different types of courses. Like if you're doing a half-day event, maybe you don't need to provide lunch. It can be quite simple, but you still might want sponsors and you know, and then if you're doing a 5-day course with accommodations and hotels and invited speakers, it's a whole different logistics. So we wanted to have kind of a series of different types of events and what you would need to do.
All right, so I'll need to log off now. Thanks very much, everybody, for having me. I learned a lot, and I'll follow up about the working group.
Thanks, Shalin.
Thank you.
Any other questions? All right, I think we'll probably end there and wish everybody safe travels in person there. And thanks everybody online for coming.